Hello, my name is Elba Valle. I'm Mexican and I just finished my PhD in August this year. I was in ETS group for 4 years, and during that time I met great people with a deep love and compromise for science. I had the chance to be involved in several projects with biology. My research was focused in the development of antibacterial skin therapies. I published 2 papers and a third one coming soon, and I collaborated in other publications. One of the core values of ETS is teamwork and collaborative working. Regarding to my research, my first paper involved the use of fatty acids and monoglycerides for the treatment of acne vulgaris. Acne vulgaris is the most common skin disorder worldwide. In this paper, the efficacy of lauric acid and glycerol monolaurate was evidenced by the utilization of biophysical and biological techniques. Fluorescent spectroscopy measurements were conducted to assess the molecular self-assembly of the antimicrobial lipids by means of the critical micelle concentration. To study how antimicrobial lipids interact with phospholipids, which are the main components of the cell membrane, supported lipid bilayers were used as a replacement for real-time evaluation of the morphological response. The concentration-dependent effect of the compound was tracked in the change of mass and elasticity and viscoelasticity of the supported lipid bilayer platform with a QCMD, which is one of the main tools used in our lab. To complete these experiments, fluorescent microscopy measurements were collected to visually observe the morphological changes. Together, all these experiments provided a deep insight of the disrupted behavior. Lastly, biological assays were performed to determine how pH-dependent conditions affect the inhibitory activities of the antimicrobial lipids against Cutibacterium acnes, which is the bacteria behind the acne disease. My second paper involved the evaluation of glycolic acid. Glycolic acid is a widely used alpha-hydroxy acid in skincare. This paper summarizes up-to-date information of the compound and provides for the first time information regarding the effect on C. acnes bacteria. The screening of the inhibitory concentration of glycolic acid against C. acnes was done with an antibacterial test named minimum inhibitory concentration. Next, enumeration assays to evaluate the concentration range at which glycolic acid kills C. acnes in different concentrations was obtained with another test named minimum bactericidal concentration. Later on, to confirm the cell killing, confocal microscopy image was performed to distinguish the live and dead bacteria. And the permeation effect of cell membrane was recorded by monitoring the release of adenosine triphosphate, ATP, which is a sensitive marker for membrane damage. My last paper was more ambitious than the first 2, and it was focused on the mixture of the monoglyceride, glycerol monolaurate with DOPC to form bicels. Bicels are cell membrane mimics made from long and short-chain phospholipids, which form 2 disc-like shaped structures. The objective here was to enhance the penetration of the monoglyceride into skin using a carrier. The bactericidal concentration, or also named MBC, of the DOPC GML bicels was ascertained to corroborate its feasibility as a reliable antibacterial treatment. The results were complemented with confocal microscopy images taken with bacteria counter-staining using a live dead kit, which is also widely used in biological techniques to differentiate live and dead cells after being in contact with any compound. A deep biophysical characterization was done to verify the membrane disruptive activity early described with glycerol monolaurate over a supported lipid bilayer, obviously utilizing again QCMD. Additionally, the possible formation of a supported lipid bilayer according to the ratio of long and short chain lipids, meaning ratio of VOPC and GML, was determined. The morphological characterization of the bicells was done with dynamic light scattering and cryo-TEM. The first one, DLS or dynamic light scattering, was to determine the apparent size and dispersity of the bicells. The second one, cryo-TEM, was to have a visual proof. However, further investigations in vivo and in situ must be conducted to validate the efficacy and safeness of these bicells. Well, I hope this brief description of my PhD research give you a glance of the amazing work done in ETS lab. Thank you very much.